Hi. Thank you, first and foremost, Tommy and all the members of the Student Seminar Invitation Committee. They told me that this is the best attended seminar in the chemistry colloquium, this Kickoff student-hosted seminar. And I thought, oh no, I'm trying to get this one right. My lab moved here just about a month ago, and we're pretty much now up and running and operating next door in Keck, and I'm hoping that people I haven't met yet, which is most people on the campus, will come and visit us over there and let us tell you about what we're doing and teach us about what you're working on. So we're definitely in the kind of trying to get to know everybody phase, which is why I thought I would start this talk with just one slide that kind of gives the big picture of what my lab does, for those of you who we haven't met. We're in a chemistry department because at the heart and soul of everything we do are chemical tools and chemical ways of thinking. But we try to apply those tools and concepts to problems in biology, in medicine, with an emphasis in the area of glycobiology, which is the biology of sugar molecules. And it's the niche of biology that I became interested in back in graduate school and have been thinking about on and off again for the last decade since then. I don't even want to count how many years that was, 25 years ago or more. So in the present, my lab has a number of project areas that involve technology development, usually with a biological application end goal and driver. For example, in the world of chemical synthesis, the type of chemistry that we're known for is called bioorthogonal chemistry, which is chemistry that doesn't interact with or interfere with a biological system, and so therefore it's a kind of chemistry you can actually perform in a biological setting. And some of the applications of that chemistry include making probes and developing strategies for imaging biomolecules in living systems, especially molecules that cannot be imaged using the more conventional biological tools like genetically encoded reporters. And we started working on this with the motivation of imaging sugar molecules, which kind of harkens back to my interest in glycoscience. But it turns out that the methods are quite widely applicable to imaging a lot of different kinds of targets in various different settings. So in addition to our own interests in imaging, in model organisms, and also in the context of infectious disease, particularly in the world of mycobacterial infection, we've also found bioorthogonal chemistry to be useful for new technologies that allow us to inventory glycosylation across the proteome. So we have an active program in developing mass spectrometry methods that allow us to find where sugars are attached to proteins and how those glycosylation sites change as a function of disease state or changes that occurred during stem cell development. We also have chemical technologies we've developed that also have this bioorthogonal flavor to them that allow us to chemically modify proteins at genetically encoded sites. And we're interested in using those methods to generate chemically enhanced biologics, like, for example, antibody drug conjugates, which are an exciting new class of anti-cancer drugs already finding success in clinical settings. And then finally, I think my microphone's kind of going in and out, which could be a battery problem, Sejin, just so you know. Get a backup. Where is she? Oh, she left. Okay, well, anybody with a battery or 9-volt in your back pocket, you might want to keep it on hand. This last square, which is a project area that is relatively new to the lab, is actually the focus of today's lecture. It's conceptually straightforward approach to what we call precision glycocalyx engineering. And as the cartoon implies, what we do is chemically synthesize molecules that look like the kinds of glycosylated proteins or lipids that you would find on cell surfaces. We find ways of attaching them to cells, and then we look for gain-of-function effects that might teach us something about the biology of the naturally occurring glycoconjugates. And so the question that we are trying to provide some answers to, and what I should really say is we're trying to provide some hints at answers, because it's early days, is why do cancer cells have altered cell surface glycosylation? And to answer this question requires, a careful thinking about the glycobiology of cancer, but also an environment, as we're discovering, that is rich both in chemistry, in biophysics, in engineering, and in medicine. And that was a major motivation for my decision to come here to this amazing university and learn from all the people, including those people in the room. So this is the student-hosted seminar at what I'm now referring to as Home Sweet Home, Stanford University. Okay. So a little bit of background, because I realize glycoscience is not something that's so familiar to people from both biological and chemical backgrounds. Cell surfaces are, in many regards, basically like data sets that are made up of sugars. And that's because most molecules that are embedded in the plasma membrane, proteins, for example, are glycosylated. So they have complex carbohydrates attached to amino acid side chains. It's also true for many of the lipids that constitute the plasma membrane. They're actually glycolipids. So they have complex carbohydrates attached to lipid chains. And all of these glycans, as we call them, create what I have come to think of as a barcode. It's a data set made up of these molecules. And it changes as a function of cells' physiology in a stereotypical way. And by the way, just as people now think of the totality of proteins as a proteome, and that proteome is a signature of a cell's physiology, the same is true for what we call the glycome. So the glycome is the totality of sugars on the surface of a cell. And they're involved in biological processes that are important to the cell. The processes that are most well understood are those that involve interactions with other cells or with the extracellular matrix, which is a great thing for sugars to do since they are determinants of the cell surface. So often when you read review articles or textbooks in the area of glycobiology, you see cartoons like this one that show glycans on glycoproteins and glycolipids interacting with receptors that might be on another cell. And there are many biological systems that follow this paradigm in which sugars play an important role. But that's probably a tip of the iceberg. And there's a lot of other biology of sugars that I think we don't even really have an intellectual framework to think about yet. And it's those frameworks that we're trying to contribute to in my lab. So there's an intersection between glycobiology and cancer that dates back many decades. And in fact, when I was a graduate student, I remember, I joined the lab of a young professor who was interested in chemical synthesis of sugars and in the biology of sugars. And he handed me some review articles on the subject of glycobiology. And in the first paragraph was the statement that cancer cells have altered cell surface glycosylation. So this has been known for many decades. And in fact, altered glycosylation is such a reliable hallmark of certain malignancies that it's used clinically in the context of pathology. So for example, when a woman has a mass identified in her breast that could be cancer, she has a needle biopsy, some of the tissue is removed, and it's sent to a pathology lab where they'll look for a variety of different markers to understand the nature of that cancer, if it is one, including the expression of different kinds of sugars, right? So we use those right now for diagnostic purposes in a clinical setting. But what's interesting and also rather frustrating is that even though the correlation between changes in the constellation of sugars on cells and cancer, even though that correlation is now widely accepted and used clinically, the functional significance of that is not well understood. So most signatures of cancer that involve changes in the glycome are a complete mystery with respect to their function. So, in other words, when these sugars change, what does that actually do for the cell that might relate to the progression of disease, if anything? Or is it just an ancillary consequence of other cellular changes, but not functionally significant in the context of disease? So two of these glycosignatures that have been associated with cancers for many decades have become of interest to the students and postdocs in my lab. And it's these two glycosylation phenotypes that I'm going to talk about today. The first is what's called mucin overexpression. So mucins are a class of glycoproteins that are very densely glycosylated in their extracellular domain. And many mucins are membrane-associated proteins that might also have some cytosolic biochemistry, whereas other mucins are actually secreted into the extracellular space. But it's this dense glycosylation that is the hallmark and the definition of a mucin. And when these glycans are so closely spaced on a polypeptide backbone, there's a profound consequence to the structure of the underlying polypeptide. And as this cartoon implies, the polypeptide is forced into a rigid, extended, stiff conformation. And the consequence of that is that mucins, they tend to tower above the rest of the cell surface molecules, kind of like the giant redwood trees in a forest made up otherwise of maples and oaks and other kinds of trees. Mucin overexpression is commonly observed on a variety of different kinds of malignancies from different tissue types. Epithelial malignancies are often have elevated mucin expression. Blood-borne cancers, leukemias, brain tumors, very, very common, independent of the tissue of origin or the underlying driver mutations of the cancer. And again, the correlation between upregulated mucin expression and cancer is as old as me, probably, which is really old. Okay. Another phenotype that has been referenced in the literature now for 30 or 40 years is the observation that many cancers are hypersialylated compared to the normal tissue counterparts. What that means is that they express glycan structures that terminate in a sugar called sialic acid, which is one of the 9 monosaccharide building blocks that we use to build our glycans, and it's often at the very end of one of these chains. So the sialic acids are shown in blue in all of these different structures, and these are structures that have been associated with cancer, either alone or sometimes in combinations, but add them all up together, and the tumors tend to have a lot more sialic acid than normal counterparts. And again, we know that this is true, but why? Why? So in the modern era, when people think about what might be the driver of a tumor-associated phenotype, I think it helps to think about the cancer as a microevolutionary process. We have a lot of different checkpoints built into our cells and also into our system, into our bodies, our immune system, for example, that protect us from cancer. So when things go wrong in a cell, 9 out of 10 times, hopefully 99 out of 100 times, we check that. The cell either kills itself, it undergoes an apoptotic program, or the immune system kills the cell for us. So for cancer to progress to disease, what that means is that the cell has evolved mechanisms to escape all of these checkpoints to the point where a mass can grow, it can disseminate and become metastatic. So what drives the selection of these glyco-traits during this evolutionary process by which cells evade all of these checkpoints? And that's how we've been thinking about studying cell surface glycosylation in the context of all the different steps that must occur for cancer to arise. For example, cells must be able to proliferate without undergoing their checkpoint control mechanisms. They might have to recruit blood vessels to nourish themselves when they've formed a solid mass. For metastatic disease to progress, cells have to detach from a solid tumor. They have to migrate away from this tissue environment and get into the circulation, a very unnatural state for cells that are accustomed to being anchored in a solid tissue. And then within the circulation, they have to avoid recognition by immune cells that have mechanisms by which they kill cancer cells. And then finally, they've got to find a distant tissue site where they can establish a niche, proliferate again, and recruit nourishment, and so on and so forth. And if a phenotype arises during cancer, I think it's reasonable to assume that it gives the cell an advantage somewhere along this process. So within that framework, what we've been trying to do is study cancer glycosylation phenotypes from the perspective of the advantage that those glycans might confer. Now, this is a biological question, and a biologist might have approaches they would take to this question, but in general, the field of glycobiology has been frustrated by how difficult it is to control glycosylation using genetics tools, right? You can knock out a gene, lose a protein, look for a phenotype. That's a classic genetic experiment. Unfortunately, these glycans are not products of single genes. So it's hard to just knock down a gene, change a glycan, and look for a phenotype. When you knock down a gene that's involved in glycan biosynthesis, you change a lot of things. You get a lot of pleiotropic effects. And this is a big frustration. And that's why we thought it might be useful to think about a chemical alternative to controlling cell surface glycosylation, which is what led to this concept of glycocalyx engineering. So again, in a nutshell, what we do is we chemically synthesize structures that emulate the cancer-associated structures. We put them on cells using very simple mechanisms. For example, we functionalize them with lipid tails that literally insert into the plasma membrane exogenously. And then, once we've decorated the cells with these structures, we can look for what phenotype they might confer on those cells that relates to a hypothesis we're testing with respect to cancer. So we have synthetic methods we've developed that allow us to build basically polymer backbones, functionalize them with sugars, and then decorate the ends of these polymers with optical probes that allow us to quantitate and follow them around on cells, and anchors that allow us to insert them into membranes either non-covalently by a passive diffusion or covalently using various kinds of bioorthogonal chemistries. So now I'm going to tell you two stories that we think have shed some light on mucin overexpression and hypersialylation using that glycocalyx engineering platform. So let's talk about mucins. If you Google mucins and cancer, you'll get tens of thousands of hits. And 90% of those will focus on a very famous molecule called MUC1. MUC1 is a member of the human mucin family, and it's associated with greater than 90% of epithelial cancers. So malignant breast carcinomas are almost uniformly high or hyper-expression MUC1. In fact, I think MUC1 is the most upregulated transcript in greater than 95% of malignant breast carcinomas. So it's an incredibly reliable marker of malignancy. It's an interesting molecule that has an ectodomain that's a classic mucin-like structure. So it's heavily glycosylated along the backbone, and the glycans are attached to serine and threonine residues at the oxygen atom of those amino acid side chains. And because of that attachment, we refer to these as O-glycans. And because there's so many of them so close together, the polypeptide is rigid and extended, and it's very, very tall. So for example, EM studies estimate that the length that MUC1 projects above the plasma membrane is about 200 nanometers. That's very tall. And just to calibrate you, a garden variety membrane protein, for example, the alpha-beta integrin heterodimer, which I'm going to come back to in a bit, is about 20 nanometers above the plasma membrane. So MUC1 towers 10 times above your average globular membrane-associated protein. It also has a cytosolic domain that interacts with proteins known to be important in signaling pathways that relate to cell survival, cell proliferation, and cell migration. And these are properties that are obviously quite relevant to cancer, right? Proliferation, migration, and survival. And so for that reason, 99% of the body of literature that studies MUC1 and its role in cancer has focused on the cytosolic domain and these signaling interactions. How many people here are cell biologists or self-classify? Just a few. One of my students. Like, cell biologists, signaling is a big part of cell biology, and it's a big focus. And I think it makes sense to shine a light on this domain of the protein and these kinds of interaction. But the truth is, there are 75 residues in the cytosolic domain and more than 1,000 residues in the ectodomain. This is an exotic structure that costs an enormous amount of energy to build, and yet nobody has a clue why MUC1 has a giant mucin domain and what, if anything, this might have to do with its association with malignancy. So this has been a mystery in the literature of MUC1 for many, many years, And I have read some of that literature and also found it kind of frustrating and unsatisfying. But I became a lot more interested when this paper was published in 2009 from Valerie Weaver's group up at UCSF. So Val is a kind of a biomechanics person who studies the role of integrin signaling in malignancy as it relates to the stiffness or the physical properties of the tumor microenvironment. So she does these sort of wild experiments where they'll take biopsy tissue from human patients and do AFM analysis and measure the Young's modulus and then correlate that with signaling pathways. It's pretty wild stuff. She did not have a history of studying glycosylation, but she had a postdoc named Matt Patzik, who at the time he was in her lab started reading literature on altered glycosylation in cancer and thought that maybe he could model what a glycocalyx could do with respect to how cancer cells interact with their extracellular matrix. And that became the foundation of this computational paper. And this was the question he asked. So it was known that cells, including cancer cells, have integrins, cell surface molecules, that are important for forming adhesions to the extracellular matrix down here. And if those integrins cluster into what's called a focal adhesion plaque, that clustering can kick off a cascade of signaling events that are important for survival of the cells, as well as other things like proliferation. So that was known, but what had never been studied is what role the glycocalyx might have in modulating these interactions. And to be honest, the speculation that was out there prior to this paper was that when you change the glycocalyx, for example, decorate it with these tall mucins, that would prevent cells from interacting with the matrix. It's like covering them, for the polymer scientists in the audience, anybody a polymer chemist? One way in the back. It's like polyethylene glycol on a cell. Okay, that's what people thought mucins were like. But what he did is he modeled the interaction where he treated the membrane as a network of springs using force constants based on literature. People have measured some of these parameters. He also treated the extracellular matrix as just a rigid substrate, an approximation. And then he tried to model the glycocalyx as a kind of squishy layer, like a mattress that you can deform if you press on it. Not as stiff as the matrix, nor even as stiff as the membrane. And then he asked the question, computationally, what would you predict to happen as you thicken this glycocalyx, which you imagine could happen if you express a lot of tall mucins, or if you stiffen the glycocalyx, also something you would imagine these mucins could impart because of their rigid, stiff structures. What would happen? And quite paradoxically, his model led to the prediction that as you thicken and stiffen this glycocalyx, you actually promote the clustering of integrins through a kinetic trapping mechanism. In other words, the membrane has to deform now for an integrin to contact the extracellular matrix. But once one of those adhesions has formed, it's that much faster for the next one and the next one to form in the local vicinity of the deformation versus outside of the deformation where there's still a distance to cross. Okay? So in other words, he predicted that a thicker glycocalyx would actually promote clustering, which then could promote everything that happens downstream of integrin clustering, which is a whole lot of stuff. Okay? And one of the things I hate most about biology are slides like this, okay, where it's just a hopelessly complicated system, so I'll break it down for you. When integrins cluster, you get a plaque. That's a focal adhesion plaque. That plaque recruits a whole bunch of things that modify a whole bunch of things, and the cells survive, okay? Who's with me? Okay. But again, this was a computational model, right? And with all due respect to the theoreticians in the audience, who's willing to admit that they're a theoretician? With all due respect, you got to do an experiment at some point, okay? And so my phone rang one day, and Matt called me up and introduced himself, and he said, I have this prediction, but I don't know how to test this experimentally. How on earth could you actually control systematically the thickness and the stiffness of a glycocalyx? Okay, chemistry to the rescue, right? So we thought, well, we can synthesize glycopolymers that look like these mucins, and since we're making them synthetically, we can control their parameters, right? Their length, maybe their stiffness, if we're really clever. And then what we can do is take cells, decorate them with our glycopolymers, and look at things like focal adhesion density and size and downstream signaling as a consequence of this decoration, okay? So this was the experimental platform that we started to pursue in collaboration with the Weaver group. So now, how do you chemically synthesize mucins, right? In a way where you could control their various parameters. Well, let's talk about what they actually look like. So here's the cartoon of the mucin, and the backbone is a polypeptide that's rich in serine and threonine residues, many of which are glycosylated with these O-linked sugars. And in this family of glycosylation, the very first sugar is always N-acetylgalactosamine, which is the blue structure, and it's always linked in an alpha linkage. And there's been a little bit of structural work that's been performed on small mucin fragments, like hexapeptides, pentapeptides, mostly by NMR, showing that the alpha linkage and the GalNAc structure actually is important because there's some interactions between the sugar and the polypeptide that contribute to its rigidity. Okay, so that's actually important. And so here's an NMR solution structure, an average structure that was published in the literature, where you can see that these green sugars are spatially distributed around a central axis, which is the polypeptide backbone, and it's fairly extended, okay? It's got a little bit of a twist to it, but that is fundamentally important for the physical properties of the mucins. So we said, all right, can we chemically synthesize molecules that look like this, but that have the dimensions that you would find in native mucins? And the answer immediately was no, okay? Mucins are big, they're long, right? Their biological mucins can be thousands of amino acid residues, but most of them are certainly several hundreds. And unfortunately, at least at the time we started working on this, there really was no method to make polypeptides and glycosylated polypeptides of that length using chemical synthesis. Okay, solid phase peptide synthesis can't get you there, unfortunately. So at that time, we thought the next best thing would be to simplify this structure to something that had all the essential features that would make it look like a mucin, but where the problem of synthesis would be dramatically reduced. And that led to the design of these glycodomain mimetics, these mucin mimetics that we prepared, in which there's no longer a polypeptide backbone, but rather a very simple polyalkane backbone. This is like version 1.0. And then we envisioned that we could hang the glycans off of this backbone through oxime linkages, and then that immediately reduced the synthetic problem to simply making polymethylvinyl ketone, very simple, inexpensive polymer to prepare, and conjugating it with sugars that we had functionalized at the glycosidic position with an amino-oxy group. So that functional group will react with a ketone to make this oxime linkage, and you can do that chemistry in aqueous environments with really complex sugar structures. It's a very mild, biocompatible chemistry, okay? So those were the kinds of molecules we made in the early days. Just to show you a more complete picture of the synthesis, after trying various different polymerization methods, we kind of settled on the reversible addition-fragmentation chain transfer, take a breath, RAFT, abbreviated polymerization method. This is amazing, beautiful chemistry that is incredibly robust. And what's so wonderful about this polymerization method is that it allows you, first of all, to functionalize the two ends of the polymers differentially. So that made it easy to put a lipid on one end and a fluorescent probe or a biotin group on the other end. And also, this is a living polymerization method that gives you very good homogeneity in your polymer sizes. So the polydispersity index of these molecules, up to 200 monomer units long, was less than 1.2, which is pretty good in the world of polymer chemistry. So we used the RAFT method to put together polymethylvinyl ketone, conjugate it on the sugars, put through a maleimide conjugation at one end, we installed a fluorescent probe, Typically, we would make our RAFT reagent with the lipid of interest and have a lipid tail on the other end. And that could be a phosphorylcholine group, it could be a cholesterol group. You know, we put a variety of different hydrophobic end groups onto these molecules and characterize them using various methods, but you can see through these TEM images that these molecules look like little rods, okay, which is kind of what we were going for, to emulate natural mucins. So for those of you who are not into the chemical details, here's the bottom line. We have a synthesis of these molecules that allows us to control the end groups with different kinds of lipid groups, clickable groups to anchor them to other kinds of synthetic surfaces. We can alter the glycan structures at will just by making different amino oxy sugars. And importantly, because it's a living polymerization method, importantly for this application, we could vary the length. So in our first pass, a former student named Jason Hudak, together with a postdoc named Camille Godula, synthesized 3 different lengths of these mucin mimetic polymers, 3 nanometers, 30 nanometers, and 80 nanometers. Remember that MUC1 is about 200 nanometers off the plasma membrane, and at the time, with this chemistry, we couldn't quite get them that long without sacrificing their homogeneity. So the polydispersity indices went way up when we got to these longer lengths. So for this initial study, we just kind of kept it kind of simple and a little bit short, but as you'll see, we learned something from this. Okay, so then in collaboration with Matt Patzik over in Val Weaver's group, we started asking questions like, what happens to cells when you decorate them with these glycopolymers, right? So the kinds of cells that we worked with were mammary epithelial cells, both human and also derived from mice. And to start with, Matt did the biological experiment, which is to take these cells and express natural MUC1 fused to the green fluorescent protein. And what he observed is that, well, you can see quite clearly that when you express MUC1 on these cells, it is not uniformly distributed across the cells. You see these dark spots where MUC1 is excluded. And what's interesting is that when he stained these cells with a marker for focal adhesions, he used an anti-talin antibody. And in that hellacious cartoon, talin was one of the things that associates with the focal adhesion plaque, okay? And what you can hopefully see by comparing these two is that where there are focal adhesion plaques in red, there is an empty space, a hole, in green. And that's because MUC1 is being excluded from these focal adhesion plaques. Turns out what we think is really happening is that MUC1 is orchestrating these focal adhesion plaques, according to the model that Patzik proposed computationally. Now, when he showed these data to his biology colleagues, they said immediately, well, you know, what's clearly happening is some cytosolic biochemistry of MUC1, because it interacts with all those things in the cytosol that are involved in signaling. And then, of course, we were able to counter, no, this is a glycocalyx effect, because look, we have glycopolymers of 2 different lengths that don't have anything in the cytosol. These are purely perturbations to the extracellular glycocalyx. And if the polymers are short, importantly shorter than the integrins, which are 20 nanometers, you can see that they're quite uniformly distributed across the cell and have no relationship to the focal adhesion plaques. But if the polymers are longer than the integrins, if they're 80 nanometers, you start to see these holes where the polymers are being excluded, and those are exactly where the focal adhesions have formed. You can see this even better in a blow-up. So here's a little region of the cell surface that's been decorated with short glycopolymers, and you can see vinculin now is the proxy for focal adhesions, and there's no relationship between these two, and here's the false-colored merge. By contrast, when the cells have long glycopolymers, you see these regions of exclusion. Those are the regions that correspond to focal adhesion plaques, okay? So all we did was put polymers on these cells, and this is what happened. That's great. Some kind of organization has occurred. But what does that mean for the actual biology and behavior of the cell? Well, the first thing we looked at was survival in a soft matrix. So it was known that for cells to survive, particularly anchorage-dependent cells, they have to be able to form focal adhesion plaques and attach to the extracellular matrix and exert tension on that matrix by pulling on it, okay? That delivers a healthy signal to the nucleus of the cell. But if you take those cells out of a stiff environment and put them into a soft jello, they die. They can't get tension. They can't get mechanotransduction from that soft matrix. So what you're looking at here are control cells that are suffering from being embedded in a soft matrix. But what's interesting is that when Matt overexpressed MUC1, natural MUC1 as a GFP fusion, all of a sudden, they're able to survive in the soft matrix, and in fact, they proliferate and form colonies. And one can quantitate these behaviors by measuring the percent of cell death. So here are control cells with no MUC1, and this is what happens when you overexpress MUC1. You get less cell death, and you also get more cells per colony. Again, is this an effect that's induced primarily by the glycocalyx? We think it is, because we can emulate that with the long glycopolymers. So we did a similar experiment. We took these cells, decorated them with the glycopolymers, and then embedded them in a soft matrix. And what we found is that whereas control cells undergo a significant amount of death, as do cells that are decorated with shorter polymers, the cells that have the longer polymers now have a survival advantage. So picture that cancer cell from a solid tumor going into a state of migration, exiting the solid tumor and trying to go into circulation. That is a low adhesion setting. That could be the point at which having elevated mucins, which can deliver stronger survival signals, could actually offer an advantage. And you could imagine how that phenotype could be selected. Now, this was a really nice preliminary data set, but we didn't do a very good job at emulating biological mucins because these are unnatural backbone structures. They're probably much less stiff than natural mucins, and they're also a lot shorter than natural mucins. And just to remind you, these are the molecules that we had made in our first set of experiments with the polyalkane backbone, but natural mucins have a more rigid, surely, polypeptide backbone. More rigid because the amide bonds have restricted rotation, okay, first and foremost. And at the time we started doing these experiments, we did not have any means of generating glycosylated polypeptides of the size that nature makes. But that changed. And it changed when Jessica Kramer joined the lab. She's a postdoctoral fellow who had been trained in polymer chemistry at UCLA with Tim Deming. And the Deming group is known for developing a transition metal-mediated polymerization reaction of N-carboxy anhydride functionalized amino acids. It's a way of making very, very large polypeptides under very mild conditions. Now, what's interesting is over the last 30 years, a few groups here and there tried to do NCA polymerizations with glycosylated amino acids, but they had failed. In fact, these failures were in the literature. And most people, when they read failures in the literature, they don't use that as the basis of a new research project, right? But no, not Jessica, because she read those papers, those failures, and she said, I think I can get that to work. And she did. So Jessica has now developed this beautiful method wherein she synthesizes a glycosylated, I think I'm losing it again, a glycosylated serine residue that's protected as this NCA, the N-carboxy anhydride, which she can then polymerize with a nickel catalyst. And the polymerization chemistry has many of the very nice features of the RAFT method in that it's a living polymerization where you can control the length of the polymer with very good precision under very mild conditions. And the polymers can be end-functionalized differentially, okay, using similar chemistries. And this NCA polymerization method is really quite amazing. So Jessica's been able to make a whole variety of structures with very nicely tuned properties. So first of all, she can control the length of these molecules, short ones, medium ones, and now really long ones. So Jessica has made glycopolypeptides that are up to 400 units in length, which is about 130 nanometers long, predicted length. So we're getting there, a little closer to the natural mucins. But also by blending in other amino acid NCAs, she can vary the density of glycosylation on these polymers, which then we think will also affect their stiffness in a way that we can modulate. So she's done some very nice characterizations on glycopolymers that are 100% glycosylated amino acid down to 30% glycosylated amino acid, and they behave very differently. I won't show you all the data because there's no time, but she took these beautiful images in collaboration with Bibiana Onoa, who's a postdoc in Carlos Bustamante's lab up at Berkeley. And Carlos developed this worm-like chain model by which, looking at AFM images of linear polymers, you can take some measurements and derive a persistence length, which is kind of a measure of stiffness. And so together, what Jessica and Bibiana discovered is that when these molecules are 100% glycosylated, they're incredibly stiff. So this is an AFM image of a single polymer, and every image in the field looks like that, just a rod, rigid rod. And this number, 208 nanometers, Jessica tells me, that is roughly 4 times the stiffness of double-stranded DNA, which we think of as pretty stiff, right? So these are like, I don't know, in the parlance of my New England heritage, it's like wicked stiff or something like wicked. Okay. And then if you kind of back down on the density of the sugars, and by blending in other kind of regular amino acids, you also back down on the persistence length, right? So you can see that now they're kind of squiggly, and here they're kind of wrapping around like little pieces of spaghetti. So this is an incredible amount of control over a physical property for a molecule of this size and complexity. And we're very excited about bringing these molecules onto cells and systematically varying this physical property to see what effect that has on focal adhesion formation and signaling. So that's kind of on the near horizon. Another technology that Jessica brought to the lab was the ability to make chimeras between synthetic glycopolymers that she makes using the NCA method and recombinant protein fragments that one can generate using, genes. And so, yeah, we think it's going to be really cool to look at what happens when you express the transmembrane domain of MUC1 and then chemically fuse it to synthetic glycopolymers whose parameters you can vary now with very high precision. And I thought I would mention, you might wonder, what's the chemical junction? That's like chemistry for the biologist. But there's a real reaction there, and I thought this was worth mentioning. The chemistry we're using to do this is popularly known as the tetrazine ligation. It's a conjugation of norbornene, which we introduce into the recombinant fragment using unnatural amino acid mutagenesis methods, and a tetrazine, which we chemically attach to the end of the glycopolymers. And these two things react with each other to form a bond very rapidly under bio-friendly conditions. It's kind of like it's a chemistry that relates to Stanford because Neil Devaraj, who's the scientist that invented this, was a grad student here. And in fact, he's coming here to give a seminar, I think in November, right? So make sure you come in here, Neil, when he arrives, because he'll tell you about the history of this amazing chemistry, which we now use. That's wonderful. But anyways, the long and the short of it is based on our preliminary studies and hopefully reinforced by experiments that are on the horizon, we now have a hypothesis for why so many different kinds of cancers end up with high levels of mucins. That property may be selected for under minimal adhesion conditions during the progress of tumors progression. And so we can now test that more directly using a variety of different animal models, taking cells, engineering them to have new types of glycocalyces, and then seeing how that modification affects their behavior in vivo, their dissemination, their survival, ability to metastasize, and so on and so forth, which we're doing with Val. Okay. So that's the mucin story, and then I have one little short story which has to do with hypersialylation. That's the other glycophenotype that we started working on more recently. And again, it's been known for a long time that cancers have a lot of sialic acid, and the question is why? Well, about 5 years ago, a paper was published by Rachel Lichtenstein's group. She's an immunologist in Israel who made the observation that if you induce tumors chemically in a mouse, they'll be hypersialylated, but if you compromise the immune system of those mice by knocking out critical cytokines like interleukin-1 alpha or interferon gamma, that hypersialylation phenotype is muted. And I won't go through these data, it's not that important, but what is important is that this paper suggested that hypersialylation might have something to do with the immune system. All right, well, when you talk to a glycobiologist about sialic acid in the immune system, they immediately start thinking about a family of receptors called the SIGLEX. And SIGLEX stands for sialic acid binding immunoglobulin superfamily lectin-like receptors, okay? And blah, 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 I know. What that means is it's a family of molecules that all share a common domain that binds that sugar, sialic acid. And then there's other domains that make these molecules different from one another. But what's interesting is that they are found on immune cells. Different kinds of immune cells have different members of the family. But most immune cells have at least one of these SIGLEC family members. And the 2 or 3 that are the most famous are indicated down here. And of those, the one that's the best studied by far is a B cell receptor called CD22 and also called Siglec2. All right, so what's up with Siglec2? This is a molecule that is known to play a role in suppressing B cell activation. So it protects us from autoimmune reactions, from reacting to our own cells and tissues, and that would, of course, cause illness. We know this because if you knock out that protein in the mouse, they become susceptible to all kinds of autoimmune conditions. The model by which CD22 acts, which has been studied extensively by Jim Paulson at Scripps and also by Paul Crocker, is by co-localizing at the synapse of a B cell and its target with the B cell receptor. Okay? So the B cell receptor is an antibody-like molecule on the membrane of a B cell. It binds to an antigen on a target, and that binding is part of a synapse that will activate the B cell. Okay? But if CD22 is clustered at that same synapse by virtue of its interaction with sialic acid residues on that target, well, that turns down the B cell. And the way it works is it recruits phosphatases called SHP phosphatases that dephosphorylate molecules that are involved in a signaling complex. All right, so we know that about CD22. What's interesting is that CD22-like molecules are found on other immune cells, and from our perspective, we were particularly interested in the observation of Siglec7 and 9 on NK cells. Okay, who's an immunologist here? One. And you're a young immunologist because you're in my group, and I know who you are. But if you were older immunologist, like if you took an immunology class 20 years ago like I did, nobody cared about NK cells, okay? People cared about B cells and T cells. That's changed a lot in the last 5 years because now we appreciate that NK cells are major players in immunity against cancer. They're really important. NK cells have these Siglec molecules that, with respect to their domain structure, are very similar to Siglec2. The most important similarity is that they have what's called an ITIM domain, this little red square, in the cytosol, and that's the part that recruits the SHP phosphatases to the synapse. So NK cells are thought to eradicate cancer cells by forming synapses and then doing a calculation. So NK cells have activating receptors and inhibitory receptors that are not all that well characterized. And when they form a synapse with a target, they're measuring, the balance between these kinds of signals. If they get inhibitory signals, they will conclude that there's nothing wrong with that cell, and they'll leave it alone. But if they get, on balance, more activating signal, then they degranulate and send out all kinds of toxic factors like enzymes and reactive oxygen species and ligands that bind receptors that induce apoptosis, and they're incredibly vicious, okay? So the calculation that they're doing at the synapse is absolutely vital to their decision making. And based on what we knew about CD22 and B cells, we wondered whether it was possible that if the tumor target was hypersialylated, right, had a lot of these sialic acids, and therefore able to recruit one of those Siglecs to that synapse, maybe that would then shift the balance to inhibitory signaling and back off the NK cell. And you could imagine how that would be an advantage for a tumor cell and how that phenotype could be selected under the pressure of surveillance by NK cells. So that we wanted to test using these glycopolymers. Now in this case, it's not so much about the length of the polymer, it's more about the structures of these glycans. And what we did is we built a variety of different kinds of glycans that had sialic acid or controls that did not have sialic acid and that were functionalized with the amino oxy group, which we were clicking onto our first generation polymers. Okay, this was before we had the NCA method. Then we took these polymers, put them on cancer cells, and asked whether they protect the cell from killing by an NK cell. Okay? So the way we do these experiments is my student Jason Hudak and a postdoc named Steve Canham took fresh blood and then isolated the NK cells from their blood. Then they mixed together the NK cells with this tumor target cells, and the tumors were either decorated with glycopolymers or not, and then they measured killing. Okay? So what's being plotted here is % inhibition of cytotoxicity. So if there's no polymers, there's a certain amount of killing that occurs. But if those cells are decorated with a sialic acid glycopolymer, they are protected to some extent from that killing. And we found that any glycopolymer that had a terminal sialic acid on the glycan structure was protective. whereas polymers that did not have terminal sialic acids were not protective. Okay, so that was an interesting observation. But does it have anything to do with the Siglex? Okay, it turns out you can block Siglex-7's ligand binding with an antibody that's commercially available, and same with Siglex-9, the other one on the NK cell. So we use those antibodies to determine whether Siglex binding was important in this protection. And now on the x-axis is percent cytotoxicity. So when the tumor targets don't have a sialic acid polymer on them, there's a certain amount of toxicity, but when you put that polymer on, they're protected. But now if you block Siglex-7 in that same experiment, they lose some of that protection. Now they become susceptible to killing. They still have the polymer, but Siglex-7 on the NK cell is blocked. Whereas blocking Siglex-9 did not seem to undo this resistance, so we think this is primarily a Siglex-7 mediated event, at least in this particular assay. And this is an isotype control which had no activity. Jason also did some microscopy and observed that Siglex-7 was in fact congregating at the synapse between NK cells and tumor targets in vitro. So in this experiment, the tumor target was a T-cell lymphoma called the Jurkat cell, and that's this big round cell. And this little thing is the NK cell. They don't look like much, but they can really kill. It's unbelievable how toxic these things are. And you can see that these two cells have formed a synapse. Now, in this case, the Jurkat cell was decorated with that sialylated polymer, and you see Siglex-7 concentrates at the interface, and this is just the false color overlay, where the green is the fluorescence from the polymer, and the red is the fluorescence of Siglex-7 using an antibody. By contrast, when the Jurkat cells were not pre-coated or not hypersialylated, they still formed synapses with the NK cells, but now Siglex-7 is not concentrated at the synapse. And in this false color overlay, the green is now CD2, which is a T-cell marker, just so you can see the membrane. So from that data set, we were able to form a hypothesis. In this case, we speculate that hypersialylation might be selected for during tumor progression under pressure by the immune system, because the more hypersialylated, the more protection they might have from a synapse with an NK cell. Turns out that NK cells also have another way of killing tumors. Again, they are incredible cells. They can do what's called antibody-dependent cell cytotoxicity. So if there are antibodies specific for a tumor cell surface antigen, those antibodies coat the tumor cell. Now, NK cells have an Fc gamma R3 receptor that binds the Fc region of the antibodies. And these kinds of synapses with Fc gamma R3 and antibodies, they're incredibly activating. So when this kind of a synapse forms, it's like bam, they just degranulate and just totally wipe out the population. And this is a very important mechanism of tumor killing with respect to the action of monoclonal antibody cancer drugs, which are now held up as kind of the paradigm of personalized medicine. For example, rituximab and Erbitux and what many people think of the great behemoth of them all, Herceptin, right? Genentech's billion-dollar drug that recognizes HER2, growth factor receptor on some breast cancers. And we now know that all of these antibodies act in part by recruiting NK cells through this kind of a synapse. This is very important. What's also true, however, is that there's a lot of clinical resistance against these antibody drugs. And for example, I was kind of shocked, given how much airtime Herceptin gets as a poster child for personalized medicine, it's not a very effective drug if you actually look at success rates in the clinic. So I just copied this paragraph from a review from a couple years ago, but highlighted are the important statements. It turns out if you're a woman whose breast cancer biopsy shows HER2 positive staining, even with that, you only have a 35% chance of responding to Herceptin. So 65% of HER2 positive breast cancer patients do not respond to Herceptin. They're naturally resistant. And then if you are responding, if you're one of the 35%, unfortunately, 70% of you later on will acquire resistance to Herceptin. You will stop responding. And despite the thousands, really tens and hundreds of thousands of people at this point globally that might have been treated with Herceptin, we don't actually know much about the mechanisms underlying the resistance. So we really don't know why it is that people don't respond to Herceptin initially and acquire resistance later, all right? So of course, naturally, we're thinking, well, if it's true that hypersialylation and recruitment of Siglex-7 can shut down the response of an NK cell in the context of direct interactions, what about in the context of antibody-mediated interactions? Is it possible that when the cells are hypersialylated and can therefore recruit Siglex-7 to the synapse, this synapse goes from being activating to being inhibitory, or less activating at the very least? And if that's true, maybe this could actually be responsible for some fraction of clinical resistance to these antibody drugs. So we tested this in our in vitro cell culture model. In this case, we combined HER2-positive breast cancer cells. It's an N87 cell line. We combined these with NK cells. We coated them, we hypersialylated them with polymers, or not. And this time we now added antibody Herceptin to the cell culture media so that we could look at the antibody-dependent fraction of cell killing, which is quite a bit. So what you're looking at here are untreated NCI N87 cells co-cultured with NK cells at different target ratios and together with the HER2 antibody Herceptin. And there's quite a bit of killing that occurs. But if you coat those cells with a sialylated glycopolymer, you can protect them from a vast majority of that toxicity, whereas if you coat them with polymers that don't bind Siglex-7, there's no protection, okay? So this is great, it's a cell culture experiment, and now I think the loaded question that we really want to answer as a group is, does this phenomenon actually have clinical relevance? And as it happens, the best person to work with to ask that question is right here at Stanford, and that's Mark Pegram, who directs the breast cancer program and was involved in the clinical development of Herceptin with Genentech and Roche, decades ago now. So he's got access to thousands of patient samples, right? Tissue blocks of tumor tissue from women who were treated with Herceptin and hopefully whose response rate we can get access to so that we can correlate whether these tissues have Siglec ligands with whether these women responded to Herceptin. And if it's true, if we can convince ourselves that this might actually be a viable clinical resistance mechanism, there are some therapeutic implications of this. For example, if it's true that hypersialylation and recruitment of Siglex-7 is protective for the cancer cell, you could imagine simply blocking Siglex-7 with an antibody. And there's one available, it's a mouse monoclonal, so we could test this in models. And you imagine co-treating the patient with the antibody that attracts the NK cell and with the antibody that blocks Siglex-7 so that the NK cell can unleash its fury on these cells, unencumbered by these negative inhibition pathways. Alternatively, you could imagine maybe developing a kind of antibody conjugate where Herceptin is now linked to an enzyme that will desialylate the glycoconjugates that are in the vicinity of the synapse. We call this a precision glycocalyx editing approach. Maybe you could actually give these tumor cells a haircut, cut off the sialic acids, and that also would relieve them from the inhibitory signaling through Siglex-7. And these are things that we're thinking about and even working on in the laboratory. Okay, my time is way over, so with that, I think I'll end and thank the people who contributed to the work I talked about today. Our first generation wave of glycocalyx engineering people were Jason Hudak, who was a Berkeley grad student. He's now a postdoctoral fellow in Dennis Casper's lab at Harvard Medical School, and he just had a paper come out on some glycocalyx engineering of the microbiota. It's pretty cool. Steve Canham, he came from a background in total synthesis of natural products with Larry Overman. Then in my lab, he kind of dove into this immunology project, and now he runs a group at Novartis in Cambridge in the kind of space of chemical biology. Camille Godula, he was a synthetic chemist who worked in my lab as a postdoc, and then he kind of morphed into more of a glycobiologist, and now he's got his own lab in the chemistry department at UC San Diego, enjoying collaborations with the famous Neil Devaraj. So they're having a good time together. Then there's the new generation of glycopolymer folks. The eminent Jessica Kramer, who brought to our lab the NCA polymerization method and is just firing out her applications on the academic job circuit this month. So thank you for sending me slides when I asked you last night, because I know you have a lot of deadlines you have to worry about. Elliot Woods is an MD PhD student who's doing his PhD in bioengineering, and he's now developing some really interesting glycopolymers that have lipid tails that allow those polymers to persist on cell surfaces for many, many days and maybe even weeks. So this is going to change the way that we think about doing in vivo experiments, which is wonderful. And then finally, Matt Zhu, who's a graduate student who's working on a completely different approach to immune oncology using glycopolymers that might dress up a cancer cell to look like a fungus. And we call that project in the lab Fighting Cancer with Dandruff. If you want to know more about that, you can talk to me. And then there's everybody else whose work I didn't have time to talk. Yeah, that includes you, Giannis. Everybody else whose work I didn't have time to talk about this time around. But anyways, I've been very fortunate to have a loyal group of Berkeley colleagues who were willing to cross over to the other side and come down here to Stanford. The entire lab came. I don't think we hardly lost anybody. And so I really do owe a debt of gratitude to UC Berkeley that provided an environment where I could recruit those kinds of people over the last 20 years. We were funded by the NIH and HHMI, but now we're here at Stanford. We're having a great time, enjoying some really cool new collaborations, and we already have a couple of Stanford students. I have a Rotan from the MD PhD program, and I even have an undergrad, which is cool. So thank you very much, and hopefully there's time for a question or two. Appreciate it.
I just had a couple questions about, first talking about the persistence length of your, essentially the peptide backbone polymers versus the aliphatic backbone polymers. Do you think is there really a big difference there, or are there other reasons that we'd be more interested in a peptide backbone versus another peptide?
Yeah, so the answer to both is yes. We think there is a big difference, but to be fair, we have not done the AFM analysis on the older generation of glycopolymers. The evidence we have that they're softer than these new glycopolymers comes more from some of the microscopy studies we did with Val Weaver's group, some turf microscopy, and looking for how they deform in the vicinity of these focal adhesion plaques. But I would bet a tiny amount of money, which is all I have after moving here to this area and buying a house. I would bet all that's left in my bank account that these polypeptides, especially the 100% glycosylated polypeptides, are way stiffer than those polyalkane molecules. That has to be the case. There are other reasons, though, to want to have a polypeptide besides persistence length. What I didn't go into is that Jessica has some evidence from some CD spectroscopy she's done that the mucins actually adopt a discrete conformation. They have a secondary structure, which is not a random coil. It's not certainly alpha helical or beta sheet. It's different from any other canonical secondary structure. So they have a real mucin fold, which could be important in ways that we don't understand right now because we don't have the framework to think about it. But there's something unique about that motif. You need a polypeptide for that.
My question is kind of related to that mucin fold, actually. When during the synthesis of mucins are they glycosylated? So are they allowed to fold and then get glycosylated? Or
oh, it's a great question. Okay, I can't answer that quickly, but I will say, here's a cartoon. So the most important amino acid for the mucin structure and the mucin fold, and this has been demonstrated in these little NMR experiments, and we've seen it now with our bigger molecules, is that very first GalNAc. Okay, so all you really care about from a structural perspective is getting that GalNAc on. There are 20 enzymes that do that in human, 20 that do that chemistry. They are situated throughout the secretory pathway. So the way that these molecules are made is it starts out by co-translationally, they get extruded into the lumen of the endoplasmic reticulum. And as they're coming into the ER, it's entirely possible that some of those GalNAcs are going on before translation has even finished. But then, since we know that these enzymes are situated all across, some are early, some are late, probably what's happening is some of the GalNAcs go on early, more go on late, even more go on later, and you kind of start with a few and then fill in the gaps, so it's regulated that way. But yeah, I think it's entirely likely that at least some of that glycosylation has occurred before you've even finished making the protein. How's that? That's a great question. If there was a way to study that, like in real time, that would blow my mind anyways, not yours.
So I have a question about the signaling inducing resistance to the receptor. So how come patients who start responding, so why are they responding? Why isn't the signaling causing them to, not respond? And then why is it like an onset?
Okay, to be fair, we don't know that that's actually happening in patients. It's a hypothesis we need to test, okay, to be fair. But, so I think maybe a question is, does it make, I went the wrong way. Does it make sense to think about patients starting out with one phenotype and then progressing to another phenotype? Is that what you're talking about? Or, so you could imagine patients that do respond to Herceptin must have a robust ADCC response. So they have robust antibody-mediated cell cytotoxicity, and which means that this synapse is a nice functional synapse, okay? So now what are possible mechanisms of resistance? Well, you know, first of all, I should say how this was even discovered is that there are polymorphisms for the Fc gamma R3 in the human population that affect the affinity of that receptor for the Fc part of the antibody. And we now know retrospectively that the responders tend to be people with a high affinity polymorphism, and the non-responders sometimes are people with a low affinity. So that right there's a mechanism that can affect response. It has nothing to do with sialic acid. But now, you know, again, cancer is a microevolutionary process. Tumors are very heterogeneous. There's a lot of epigenetic changes that are occurring, right? And these things are happening under conditions of selective pressure. So, you know, it's possible that hypersialylation has other benefits to the cell outside of this NK response. And it's just an added benefit that confers resistance to this mechanism. So we can't rule that out. But since no one's actually ever looked at the glycocalyx and correlated glycosylation with susceptibility to these drugs, we have no idea where to start, right? You gotta start somewhere. So,
do you think it's a phenotype that's induced or like evolutionarily gained?
Probably it's an epigenetic. It's probably gained epigenetically. I think that's where the smart money is. You know, it's, for example, many of the tumors that are hypersialylated on mucins. So you could imagine that, you know, you acquire a hypermucin phenotype first, that confers its various benefits, and then when you acquire sialylation on these mucins, or hypersialylation, you have an added benefit of immune surveillance, you know, escape. You could imagine, right? Speculation, though, to be fair. Right? Be cool if this is true. Like all of our projects, yeah.
So I'll preface this by saying, of course, I know absolutely nothing about immunology, but it seems to me that in the model where you're proposing to study this by inhibiting Siglec-7 to see if basically you can induce these NK cells to degranulate and destroy everything, doesn't that seem maybe a profoundly dangerous way to go about fixing the problem?
Well, let me just tell you something. So you're saying, like, are you kidding? Why would you want to block Siglec-7 and then hyperactivate the immune system against tumors?
Well, what I'm actually saying is if every cell at some point is interacting with an NK cell, and then you turn off or reduce its ability to, you know, mediate that interaction, might you not instead end up destroying your body rather than targeting your?
Well, okay, yes. Having said that, you know, there are some very exciting developments in the field of immune oncology from the last few years, including 2 big drug approvals in which co-administered with the chemotherapy is an antibody that blocks a T-cell checkpoint. And have you heard of these PDL, PD-1 and PDL? Merck has a drug on the market now, Bristol Myers Squibb has a drug on the market, and what they're basically doing is taking the brakes off of T cells while they're administering a chemotherapy. And the consequence of this is you get much stronger T cell reaction to the tumors while you're also killing them, and this leads to a more durable response against the cancer. And side effects that are autoimmune in nature, okay? But, you know, that's life when you're a cancer patient. You're going to have to deal with side effects. And I think it's a fair argument that the autoimmune side effects that occur during treatment with these checkpoint inhibitors are not as dangerous as some of the, you know, side effects of higher levels of chemotherapeutic drugs or second-line treatments with chemotherapies because the first line was not durable. So, you know, you got to make choices when you're treating any disease, certainly cancer. But I think temporarily suffering from autoimmune side reactions that's a price most cancer patients are willing to pay, who have very few alternatives. So I don't think there's anything outrageous about proposing to shut down Siglec-7 while you're treating a patient with other drugs. It's just a different branch of the immune system compared to the T cell pathways that people have already targeted with great success in the clinic. talk about that. Okay. So the mucins, the natural mucins, or both, and ours too. Okay. So actually MUC1 is a very kind of like dynamic molecule. It has an internalization signal in the cytosolic domain, and it's constantly being internalized. And nobody knows why that is or why it's important. We have some ideas now in light of these models with the Weaver group and so on, but it's a dynamic molecule. Of course, always new molecules are being made, so you have a constant supply of MUC1 through biosynthesis. By contrast, when we put our synthetic molecules on the cell surface, they too are going to turn over because membranes are constantly internalizing, and there is no new supply of them unless we add more, but you can't do that in the models that we're trying to study in vivo, for example. Which is why it was really important, actually, that Elliot was able to identify a lipid structure that allows these molecules to persist for a long time on the plasma membrane. And it really is all about the lipid here, it turns out. We just have discovered recently. So what he found is that, he just submitted a paper on this today, that cholesterolamine, it's not a natural lipid to our knowledge, it's a derivative of cholesterol, but cholesterolamine with a positively charged amino group, if that's the lipid anchor, what happens is these things, they internalize into some kind of compartment that co-localizes with transferrin receptor, whatever that means, and they form a depot in these compartments. And we can see this from fluorescent staining, right? Just big, bright spots. And from that depot, they now get redelivered to the plasma membrane, and they just recycle from this depot. So any one of these cholesterolamine glycopolymers is moving. It's coming in and coming back out, but on bulk, they're persisting on the plasma membrane, which is amazing. What's also cool is that so much of it is in this depot inside the cell that when the cells divide and form two daughter cells, the depot gets distributed amongst the daughters, and the daughters are doing this. So the cells are dividing and dividing and dividing, but they're not losing their steady state level of these glycopolymers for a long time. Eventually they do, but it's quite cool. So that's what I can tell you. And what use we make of that, we'll be seeing. Dick.
What a wonderful thought. I'd like to follow up with asking about lipids. We know that lipids also change between cancer and normal cells. What do we know about glycosylation of lipids?
What do I know is not that much. I don't know that much about it myself. There's a couple, glycolipids are like, it's the Bermuda Triangle of biology, because like lipid biology is really hard, right? Glycobiology is really hard, right? Cancer is hard. Then you put them all together and no one has a clue. And, but so there's a couple of groups that have looked at these changes in glycosylation of lipids. There's some famous glycolipids like GloboH, right? That are hallmarks of breast cancer. People are trying to vaccinate against them. But I think what little we know about the glycoproteins, we know even less about the functional relevance of the glycolipids. Some scary stories are out there about how these glycolipids interact in cysts with receptors and modulate their signaling properties. Good luck studying that. I mean, if you have a good experimental system to study things that occur like that in a membrane, you own the world, I think, because there's a lot to discover there. And so, I don't know, I think it's like the Wild West. Glycolipids is like, put on your cowboy hat, get some guns, and go out there and take a shot at something, It's a great area for a bold student, Yeah.
So these sugars are quite a bit longer and look quite different than normal sugars on a certain small. So you think there's a synthesis of cells. Do you think that there's anything we can use to bind them to block their interaction rather than blocking the receptor on the inside?
So you're saying, could you, instead of, kind of plugging up this part, make an antibody or some reagent or something that would kind of plug up the sialic acids? Something like, yeah, you could think about that. And I think the key there would be to figure out how to target it to the tumor cells. But, maybe you could create a construct of that type. That's a great thing to think about. Yeah. I mean, our idea of hooking an enzyme to the antibody, just cutting them off, comes from the fact that these enzymes are available. People have studied these kinds of enzymes. You could imagine making the conjugates and stuff. But if you could, and they're catalytic, an enzyme can chop off lots and lots of sialic acids for one enzyme, whereas a blocking agent, you have to figure out how to block a lot of things stoichiometrically. But that doesn't mean it can't be done. It's a good thing to think about. Okay. Thanks for hanging in there with me. Appreciate it.